Mrs H here. This video will take you through some practice exam questions. We start off with easier questions and they get progressively more difficult towards the end of the video. The image shows plant shoots growing towards sunlight. So any movement towards a stimulus is positive. In this case, the growth is towards light. Well, photo is light, so this is positive phototropism, so the answer is D. Plant roots respond to gravity by growing downwards. What is this response called? Well, they're growing towards gravity in the same direction, so that is a positive, and it must be positive gravitropism, so that is C. Which hormone is important in the fruit ripening process in plants? Well, that is ethene and that is A. Now, those hormones are naturally released, but what we can do is if we extract the ethene, pop it into a spray, we'll pick our fruit unripe, then we spray them while they're in transit, and then we can sell them nice and ripe. So that's how we utilise these natural hormones in an artificial way. Question 4. The diagram shows apparatus used in experiments on tropisms. Here we can see that a seed has been pinned to a cork board. We can see the root is breaking out of the seed. Normally roots grow downwards towards gravity because of the effects of auxins collecting on the underside, inhibiting root growth on the underside. That would mean that the upper side will elongate more than the underside, causing the root to bend downwards. But in this piece of apparatus, if we keep rotating the seed, then the auxins cannot collect on the underside. So you don't end up with an uneven distribution of auxin. So no side elongates more than the other, and the root ends up growing horizontally instead of downwards. We are removing the effect of gravity that normally causes auxins to collect on the underside. So we are removing the ability for positive gravitropism, which is B. In 1913, Boyce and Jensen also investigated phototropism. The diagram shows his results. So we can see here we've got two shoots. They've had their tips cut off and then they've had their tips stuck back on. But in the diagram on the left, the tip is separated by the rest of the shoot via a gelatin block, which allows diffusion. And the shoot on the right, the tip is separated from the rest of the shoot by a mica block, which prevents diffusion. So the question is, explain how these results show how auxins move. Well, auxins are made in the tip and they diffuse to areas just below the tip. And these areas are where cell elongation can take place. So where we see growth is happening towards the light is because the auxins have been able to diffuse to the elongation zone where they cause the cells to elongate. And as we know, auxins move away from the light, so will gather on the shady side, causing elongation of the cells on the shady side, which causes the shoot to grow towards the light. In the shoot growing straight up, the auxins remain in the tip. They can't diffuse downwards to the cells in the elongation zone, so no elongation occurs. So what was the question again? Explain how these results show how auxins move. Well, the actual answer is auxins move by diffusion. How can we tell? Because when auxins cannot diffuse, like on the diagram on the right, there is no growth towards light, but when they can diffuse, elongation and growth towards the light can be seen. Plant development is controlled by hormones. Describe one effect of gibberellins and one effect of ethene on plant development. Well, gibberellins have a few roles, but the main one we should remember really is that it breaks seed dormancy. So in other words, it causes a seed to germinate. We can use these hormones to our advantage because if we extract the gibberellins, then we can use them to make seeds germinate when they shouldn't normally be germinating. So that's something that we do in industry. And other effects you might know about for gibberellins are that like causing fruit growth and shoot growth, but we'll stick with the seed germination. Ethene, we already know that stimulates fruit ripening, and as I've mentioned before, we can extract this, we can put it in a spray, and we can spray unripe fruit, so when it's ready to sell, it's nice and ripe. Question 7. Some students investigated phototropism in plant seedlings. This is the method used. Measure the lengths of shoots of 20 seedlings. Set up 4 groups of seedlings as follows: A, B, C, D. A, bottom of shoots covered in aluminium foil. B, the tip is covered in aluminium foil. C, the tip is removed. And D, there is no change made whatsoever to the shoot. Put the seedlings in a cardboard box. Use a lamp to shine a light into the box through the hole on one side. After 1 day, remeasure the lengths of the shoots. Make a drawing of the appearance of one seedling from each group. And here we can see the appearance of the seedlings at the start of the investigation. Which 2 conditions should the students have kept constant for each group of seedlings? Tick 2 boxes. For each group, that's what we need to focus on here. So when we're looking at group A's, group B, group C, group D, what are we keeping? What do we have to keep the same between A, B, C and D? It's a bit of a trick question, this one. The length of the roots, well, we can't control that. The number of seedlings in each group, that's already been decided. We've been told we've got to have 20 seedlings. There are 4 groups, so 4 different conditions we're investigating. That means we're going to have 5 sets. So we can't change that. The temperature, yes, we need to make sure that we keep the temperature the same for all seedlings A to D. Thickness of aluminium foil. Well, only A and B have foil, so we can't treat them all in the same way. If you were looking at all the groups in the repeats, then yes, the thickness of the foil for A and B needs to be the same for all of your repeats. But when you're looking at comparing A, B, C and D with each other, they're not all got foil on the top, so that's pointless, so that's not an option. The volume of water added to the soil, yes, we need to make sure A, B, C and D have the same volume of water added. What is the purpose of aluminium foil? To hold the shoot straight, to keep the shoot warm, to remove the effect of gravity, or to stop the light reaching the shoot? It is to stop light reaching the shoot. So here we can see the students' results, and we've got some data in the table. Suggests how students measured the lengths of the curved shoots of seedlings A to D at the end of the investigation. Well, it says to suggest, so anything that you think is feasible could get you a mark. But the best idea would be something along the lines of using a piece of string, placing it along the length of the shoot, because it is curved. You can't really use a ruler on a curved shoot. And what you can do is mark the start and the end point. Then you hold this piece of string against a ruler, and then you have your measurement. A hormone stimulates growth in shoots. Which distribution of the hormone would cause the results seen in shoot D? Tick 1 box. We know light causes auxins to move to the shady side of a shoot, and when they do gather on one side, they cause elongation on that side, in shoots anyway. It definitely isn't the middle one, because auxins are in an even distribution, So there won't be any elongation on either side. The shoot will just continue to grow upwards in a straight line. If it was this top one, then that would cause this side to elongate and the shoot would actually bend this way towards the left. So the answer is this one, definitely the bottom one, as the shady side will become longer than the lighter side and the shoot has to bend. A student investigated the effect of a plant hormone on the growth of roots by plant cuttings. The student took 6 stem cuttings from the same plant. He put the cuttings in test tubes containing hormone solutions of different concentrations. The image shows 6 cuttings after 2 weeks. Okay, what is the best concentration of hormone for encouraging root growth? Wow, wouldn't we love this one in an exam? That's definitely 3, isn't it? That's nice and easy. Give 2 functions of plant roots. Okay, absorb water from the soil, absorb minerals from the soil. You might also put anchorage and support, but remember, you just got to write 2. Draw a ring around the correct answer to complete the sentence. Taking cuttings to produce new plants is an example of asexual reproduction. Another student investigated the effect of light shining from one side on the growth of plant shoots. The diagram shows how the student treated the shoots and the results she obtained after 12 hours. What is the response to light shown by seedling A called? Cloning, a reflex, a tropism. A tropism, lovely. The student concluded that the shoot tip is sensitive to light. What evidence is there in the diagram above for this conclusion? The important part to notice here is the evidence from the diagram. The tip of seedling A is exposed to light and it grows towards the light. That is the evidence from the diagram. The tip of seedling B is covered and it does not grow towards the light. The seedling produces a hormone which helps to control its response to light. What is the name of the hormone? Auxin gluten. Okay, it's definitely auxin, isn't it? How does the hormone control the response of seedling A to light shining from one side? Well, the auxin is made in the tip and moves to the shady side of the shoot. This stimulates the shady side to grow or elongate more than the other side, and that causes the shoot to grow towards the light. A student investigated the effect of a plant hormone on the growth of roots by plant cuttings. The student took 6 stem cuttings from the same plant. He put the cuttings in test tubes containing hormone solutions of different concentrations. So this shows experiment 1. The tip of the shoot was removed from the plant and there was no growth in the shoot after 3 days. Experiment 2. The tip of the shoot was cut off and then placed back onto the shoot. The shoot had grown 6 millimeters after 3 days. Give 1 variable that must have been controlled for the plant shoot to grow vertically, as shown in experiment 2. Well, we need to make sure then, if we are going to make sure the auxins are evenly distributed, the direction of the light needs to be even on all sides. So the direction of light, or ensure equal light intensity around all of the plant. State 1 conclusion that could be made based on these 2 experiments. Well, the substance, auxin, that causes shoot growth is found in the tip of the shoot. It must be, because when you take the tip out, it doesn't grow. Which plant hormone causes growth in the plant shoot? Auxin. Explain 1 way that this investigation could be improved. Well, the best one that you could say here is that you can repeat the experiment with more plant shoots. And the reason you want to do that is because you need to identify anomalous results. So identify anomalies. then you can remove those anomalies when you calculate a mean, and that will give you a more valid answer. And this is the final question. Some students investigated geotropism in the roots of bean seedlings. Geotropism is the growth affected by gravity, and we know in roots grow towards gravity, and in shoots above the ground they grow against gravity, don't they? So they grow upwards, roots grow downwards. Apparatus A, we can see that the seedlings have been pinned to this cork mat, and this apparatus is kept stationary, but an apparatus B is attached to a motor which rotates. So those seeds are rotating around all the time. This is the method used. Measure the length of the root of 10 bean seedlings. Pin 5 seedlings to the cork mat in apparatus A. Pin 5 seedlings to the cork mat in apparatus B. Leave A and B in a dark cupboard for 2 days. After 2 days, make a drawing to show the appearance of each seedling and measure the length of the root of each seedling. Okay. Why did the students surround the seedlings with damp blotting paper? Tick 1 box. To prevent light affecting the direction of root growth. Well, it's not that one, is it? Because if you think about it, roots grow underground, so they do not depend on light anyway, so it's not going to be that one. Is it to prevent photosynthesis taking place in roots? Well, photosynthesis doesn't take place in roots, there's no chloroplast there, and like we said before, they don't need light. To prevent growth of mould on the roots, I'm not sure how that's going to help. If anything, that would encourage it. And to prevent water affecting the direction of the root growth, yes, because as well as geotropism, roots grow towards water, don't they? So if you're completely surrounding the seed in the dark and completely surrounding it with water, there's no preference for the root to grow towards. Apparatus B is a control. Apparatus B rotates slowly. How does apparatus B act as a control? Well, we're trying to investigate the effect of geotropism, which is growth, and the effect of gravity on growth. So B is rotating constantly, so it's cancelling out the effect of gravity. So we get to see how seeds grow when gravity is not having an effect and when it does have an effect. 6.3. One student stated the mean length change for the seedlings in apparatus B is not valid. Suggest the reason for the student's statement. Well, quick, have a look. The mean actually includes the result for seedling 4, which is anomalous if you look at all the rest of the data around it. Suggest one improvement the students could make to obtain a more valid mean length change for the seedlings in apparatus B. You just remove that anomalous result and calculate the mean without it in there. Or you could repeat the entire investigation and calculate a new mean. 6.5. Figure 7 shows the students' drawings of 2 seedlings at the end of 2 days. So the seedling from apparatus A is growing downwards, and the seedlings from apparatus B is growing fairly straightish. A plant hormone is made in the root tip. That's our auxins. The hormone diffuses from the tip into the tissues of the root. Explain how the hormone causes the appearance of the seedlings to be different. You should refer to both seedlings in your answer. So the seedling from apparatus A, the auxins move towards gravity and will be found on the underside of the root. This causes the bottom to elongate less than the top. So the fast elongation at the top of A makes the root grow downwards. And if we have a look at the seedling from apparatus B, the effect of gravity is removed by the constant rotation. This means auxins do not settle on the underside, therefore creating an even distribution. So elongation will occur at the same rate on the top side and the underside. So you haven't got one side of it elongating faster than the other. So there's no bendiness that's going to take place. And 6.6, in horticulture, plant hormones are used for controlling plant growth. Draw one line from each plant hormone to the correct use of that hormone. Auxin, to stimulate root growth in plant cuttings. Ethene, to reduce the time taken for tomatoes to ripen. And gibberellin, to promote seed germination. Thanks for watching. If you found this useful, please like and subscribe to keep up to date with more content.